Hello students, myself Dr. Luqman. I am here to teach you about NEET revision video series. In my introduction and also in my first video, I have started the series. Now I am going deep dive into the exam oriented high scoring topics. In my previous video, I have taught about first chapter, the living world. From that video, minimum 4 marks are expected and maximum 8 marks are expected. And even if you find that video to be of lesser important, only 4 marks or 8 marks, but majority of the terms and definitions are actually the foundation for the other videos. In this video we are going to teach about biotechnology. We are going to have complete exam notes. This is the only video you will need for your exam. इसके अलावा कोई और वीडियो, उसके अलावा कोई और नोट्स भी आपको पढ़ने की जरूरत नहीं पड़ेगी. Hopefully in this video we will be covering biotechnology principles and process chapter 9, and also the biotechnology and its applications chapter 10 in a single high yield exam coded document. It is every line is from NCERT, and previous year questions of nearly more than 20 years I have scanned and put it here, and have made the notes integrated and inclined with the previous year questions and examiner's mind. This is the ultimate resource for the students of MBBS aspirants. And not just about this video, after watching this video completely, I guarantee you 30 marks minimum are confirmed, because from these biotechnology and its applications nearly 6 to 7 marks are expected. Sometimes the score has been 8 questions also, so 6 to 7 questions or 8 questions, it means nearly you will be scoring 32 marks. Minimum 30 are confirmed from this one single video. I'll guarantee you, you shall be ready to solve any question, any NEET question from biotechnology. Revise these notes twice, first for understanding and also second for the memory anchors. Practice 100 plus MCQs. I myself have given in this video more than 50 MCQs from each chapter. Aim for 360 by 360 in biology, and that is this by this video you are towards achieving that number. So this is our second video. First video was about the living word, and the second video, because it is a high scoring area, my second video is about biotechnology. So let's start. Biotechnology principles and process. Let's have a examiner's blueprint. एग्जामिनर्स ब्लूप्रिंट का मतलब होता है कि इस टॉपिक से, इस चैप्टर से एग्जामिनर्स इससे पहले कितने क्वेश्चंस लिया है, और फिर अभी आगे जाके वो कितना एक्सपेक्टेड है, इसका वेटेज क्या है, कितने नंबर ऑफ क्वेश्चंस आएंगे, कहाँ से आएंगे और वो कैसे आएंगे. All these is called as examiner's blueprint. So most of the questions from this are these I have enlisted here. You can see restriction enzymes, right? Like naming the enzymes, recognition of those enzymes, cutting, sticky and blunt ends. We are going to study this. I'm just telling you the blueprint. And from this topic, the most common type of question which is expected is statement type. Okay, like true or false statements, palindromic sequence and identification, and echo-rai features. We are going to have a clear understanding of each and every topic here. We are going to practice previous year question, and also we are going to practice the expected questions. So do watch this video till the last of these slides, because this is very important. I have taken like minimum 4 to 5 hours to make you these notes, because there are many notes available, but what I have done is I stick to NCERT, but only taking out the direct exam orientation points, no other story or other introductory terms and etc. From the cloning vectors, this is also high yield point. The expected questions are from PBR 322, ORI, and selectable markers, insertional inactivation. Diagram based questions have been asked from these cloning vectors topics. Match the following questions are expected, and restriction sites within the TET-R and AMPR. TET-R and AMPR is also expected. We are going to study what are all this. Don't worry, these words might be looking shortcut, but I'm just giving you the topics from where the examiner is going to give you high yield questions. This is examiner's blueprint. If not, we are going directly to study these topics. Okay, gel electrophoresis principle, ETBR and UV elution sequence, correct or incorrect statements. These are the type of questions expected from PCRs like polymerase chain reaction. We are going to study steps, stack polymerase, amplification formula, sequence formula, application, etc. Competent host preparation. We are going to study what are the methods by which we are going to make it competent, and what is the preparation methods, CSCL2 method, heat shock method, ballistics method, and micro injection method. These are also expected questions. And from the topic of competent host preparation, we are going to expect direct fact recall questions. Downstream processing and bioreactors have been also a very important topic for the examiners. Let's go into the topic. Principles of biotechnology. Okay, let's see what is biotechnology. Definition. Even the words also are self-explanatory. Biotechnology. It means we are going to understand biology to the levels of its biomolecules and genes, so that we are going to apply the technology towards the level of biomolecules, and we are going to bring some changes. That is biotechnology, if you are going to just like break down the word also. But let's see, integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. If an examiner wants to distract you, he can give you the question on definitions also. Okay, and almost all definitions can fit in biotechnology, because biotechnology is such a vast area. So you have to stick to exact meaning and comprehension what he is asking, like from the natural science, from the organisms, from the cells and their parts, we are going to have molecular analogs, and for products and services or therapies, etc. Moving on, two core techniques that enabled modern biotechnology. Even not just now, it has been like more than 30 years, 40 years that the biotechnology has changed at a highest magnitude. The technology has changed, the methods, the processing that has affected every single part of our life. So there are two core techniques because of which this modern biotechnology has enabled into this vast magnitude. One is genetic engineering, direct manipulation of DNA or RNA, genetic genes. Okay, we know that genes are present in this DNA and RNA to alter the phenotype of the host organism. So, what is phenotype? We are going to study. I think you might have studied in the Mendel's chapter also. Phenotype is physical appearance of an organism depending upon the presence of that gene. So, a gene's physical expression is called as phenotype. So, we are going to manipulate the DNA or RNA in order to change the physically appearance of the host. That is called as genetic engineering. And the second technique which has enabled this biotechnology to its vast field is bioprocess engineering, maintenance of sterile microbial contamination free conditions in large scale culture to produce antibiotics, vaccines, and enzymes is called as bioprocess engineering. Sometimes direct definitions are being asked. So remember this definition. You can see this diagram also, which can clearly depict what is genetic engineering and also this bioprocess engineering. That can, by seeing the images, you can remember these definitions very easily. Why genetic engineering is superior to traditional hybridization? First of all, like traditional hybrid, let's understand what is traditional hybridization and what is genetic engineering. Okay, let's compare it. So in traditional hybridization, what happens is we are going to just have a natural observation of these organisms, let's say plants. Okay, in traditional hybridization, it uses entire organisms, and natural crossing will happen, like natural crossing, natural breeding will happen. But in that one, crossing and after hybridization, the progeny, it means the next generation desirable organisms or plants or animals, they are going to have both the traits, one which we desire to do that hybridization, and the other which we don't desire. Undesirable genes also come along with the desirable ones. And whereas in genetic engineering, we are going to have an isolated approach. We are going to have isolation and introduction of only one or a set of a desirable genes without any undesirable genes. The picture beside that, you can see there is a specific understanding what is the difference between traditional hybridization and genetic engineering. You can see that, okay? uh, Pause the video here, observe the whole topic, also observe the key features and note it down. That is what is important for understanding. Fate of alien DNA in a host. Important previous year question. It has been asked multiple times, and expected questions are from this one. Okay. An alien piece of DNA cannot multiply by itself in the progeny cells. Whatever cells, progeny cells, if an any alien means foreign, undesirable, okay, which we did not expect, something from outside the natural environment. So this alien DNA cannot multiply by itself in the progeny cells. It must be linked to an origin of replication, ORI, origin of replication, in order to replicate. Once integrated into the host genome, it may multiply and be inherited along with the host DNA. It means if you just any alien DNA is getting inside the cell, that DNA is directly not going to replicate. Okay, one has to have an exact origin of replication, and from that point only, if it is integrated to the host cell level, then only it can multiply and the character can be inherited along with the host DNA. You have to understand this one. One concept is alien DNA has a limitation. Alien DNA cannot multiply by itself. We can see the diagram and remember, because the more you imagine process, it will be easier for you to understand also and also answer the questions. If not, all these topics can be like vanished from your memories, or you might get confused. Imagination is always key to success. Okay, and the replication, what is required for the replication, it must be linked to an organic to the origin of replication, that is ori, then only it can have its replication. Okay, may multiply and can be inherited along with the host DNA after the ori only. Examiners trap, see here, a direct pyq has been asked. It means previously a question has been seen in the NEET. What is the fate of a piece of DNA carrying only gene of interest transferred into an alien organism? Okay, he has just prolonged that sentence to confuse you. Obviously, it may be integrated into the genome and multiply inherited, but it does not multiply independently without ori, that is origin of replication. It cannot multiply on its own. So you have to remember that. Moving forward, this is one of the very important topics. Remember the image. That image will help you a lot. The first recombinant DNA, genetically modified DNA. Okay, first time it was done by Stanley Cohen and Herbert Boyer. Rarely scientist names are important. Most of the time we have seen and we have known that scientist names are not asked, but you cannot say. One cannot be always sure. Better be on the safer side. So first recombinant DNA was produced by Stanley Cohen and Herbert Boyer in the year 1972. They isolated an antibiotic resistance gene from a plasmid of Salmonella typhimurium. So from a bacteria called as Salmonella typhimurium, they have uh, isolated a antibiotic resistance gene called as it's a form of from a plasmid, and then they used a restriction enzyme, a molecular scissor, to cut it. Used a DNA ligase to join the gene from the plasmid vector. The recombinant plasmid was introduced into E. coli, which then replicated the gene, cloning of antibiotic resistance gene. You can see in the diagram also, the first recombinant DNA was produced in 1972 by Stanley Cohen and Herbert Boyer. Gene cloning technology. First is gene isolation from salmonella. Even though this is a topic of a history, okay, that is genetic history, what happened in history and when it was discovered. But what did they do in order to discover that process can also be sometime asked in question as part of a sequence. Okay, for that one, they done first is gene isolation from salmonella, then digestion through molecular scissors, that is cutting, then ligation through DNA ligase, then into the transformation and cloning. That's how they produced a first recombinant DNA. You can see antibiotic resistance gene was first isolated from a plasmid of Salmonella typhimurium. Then they used the restriction enzyme, also called as molecular scissors. These restriction enzymes are going to cut at the desirable location of that DNA molecule. Then they are going to use it to attach it to the actual plasmid vector through DNA ligase, and afterwards they are going to have that into a transformation and cloning will be done. So remember the first recombinant DNA, it is very important topic for your examination. Three basic steps in genetically modifying organisms. Remember, dear students, this video is all you need for biotechnology. Apart from this video, don't go into any other notes or any textbooks, anything. Nothing is required. You just need this video to understand and also revise it. Minimum watch this video into 2x speed, like 3 to 4 times. Watch first time only in a normal speed, and after second time or third time you can watch into second speed. Then you will remember the topic also. Your PYQs will be also solved, and expected questions will also be solved. Examiners trap also you will be identify that one, and the images will also be very memorizing in nature, and that will help you in the exam. Okay, the three basic steps actually in genetically modifying an organism is identification of DNA with the desirable genes. First of all, let's understand genetically modifying organism, GMO. Okay, the word itself is self-explanatory in nature. Okay, an organism which we have modified, we have changed, we have made it special. What through whatever we wanted to happen, we made that happen through that desired gene only. Such a modified organism is called as genetically modifying an organism. So first step is we have to identify the DNA that is desirable genes, and introduction of identified DNA into host, maintenance and transfer of introduced DNA to the progeny, and that's how we are going to produce the GMO. You can see there in the diagram also, we are going to do this all as a lab activity. That's the reason the diagram is like a lab type approach. Okay, first identify the desirable genes, then we are going to cut those desirable genes, then we are going to have introduction, then the maintenance and the transfer progeny. That is how it will be successfully formed genetically modified organisms. Tools of recombinant technology. We are going to study this one in detail. What do you mean by tools? Tools means what are the equipments or what are the things which we need in order to produce recombinant DNA, a combined DNA, a special DNA, a DNA with a desirable gene, and a DNA who is ready to join with another host organism's DNA, and a DNA who is going to replicate. Such type of thing is called as recombinant DNA. So in order to produce recombinant DNA also, and in order to have that whole technology, the whole process to be covered, we need a tool. Okay, so there are many tools we are going to study one by one. Okay, first is restriction enzymes. This is very important. Always minimum one question is asked from the restriction enzyme itself. Sometimes two questions have been asked within these restriction enzymes. Okay, restriction enzymes, also called as molecular scissors, the historical landmarks. Okay, when it comes to understanding the science, obviously not always history is important, but history is also important because it will make you curious how the scientists have thought about it, so that our mind also will think about the further concepts related to that science topic. So that is very important. Okay, historical landmark. 1963, two enzymes isolated from the E. coli. E. coli is a favorite topic of all the examiners. One of the most common names you are going to get in the whole biology and integrated questions, not only the questions of E. coli from the biotechnology, we are going to have integrated questions of E. coli from biology, from the protozoa, from the tissue, and other type of topics also. An integrated question only with the word E. coli can be expected. Okay, so two enzymes were isolated from the E. coli. One, methylated DNA modification. The other, cut DNA, that is restriction endonuclease. So these two enzymes were isolated from the E. coli. Hind2 was the first restriction endonuclease. Okay, so the first restriction endonuclease is Hind2 that recognized a specific sequence, six base pair sequence. It always cut at that specific point only. So historically, this we have to remember. Fact-based questions are expected from this one. Naming convention, critical for NEET, it is always important. First letter genus name capital italics. Next two letters species name lowercase italics. Remember, first letter is also in italics and the second is also in italics. But in the first letter there is genus name and capital, whereas in the second it is lowercase. Okay. Then strain designation is always in the capital letter. Roman numerical order of discovery from that strain. Such type of naming will be given to these type of enzymes. Okay, so example is E. coli. In this E. coli, that is Escherichia coli RY13, first endonuclease isolated. So Escherichia coli RY13 is the first endonuclease that has been isolated. Okay, so you can see the overview of the genetic engineering and molecular scissors. Identification of DNA with desirable genes, introduction of identified DNA into host, maintenance and transfer of introduced DNA to progeny. Then the restriction enzymes comes molecular scissors. That how does they work? Dual enzymes from E. coli. Okay, one methylates self DNA, other cuts the foreign DNA. Okay, and Hind2 is the first restriction endonuclease. Okay, and here it is, first endonuclease is Escherichia coli RY13, and the first letter next to two letters, then strain, then Roman numerical that has been given in this image. Previous year question: what does Co in ECORI stand for? It stands for coli, coli that is indicating Escherichia coli. Which restriction endonuclease was the first to be isolated? Hind2. You remember, you should remember, this is important question. True or false questions could be also asked. Match the following could also be asked, and historical importance is always one has to remember. Okay, and the diagram also shows what is the function. We are going to study that in detail later on. Okay, classes of nucleases: exonuclease and endonuclease. Exonuclease removes the nucleotides from ends of the DNA. Endonuclease makes the cut at specific positions within the DNA. Okay, so what does exonuclease do? Exo means outside, external. So removes the nucleotides from the ends, outside, last part, ending part of the DNA. From that part, this exonuclease is going to remove the nucleotides, and endonuclease is going to have cut at the specific locations within the DNA. You can see always exonuclease and endonuclease start from like 3'5" direction and 5' 3' direction. Okay, we are going to study that one also. Those are basics. Today in this video we are directly focusing on the exam orientation points, direct statements or direct word to word questions, statements in the questions could be expected. Recognition sequence palindromes. Palindrome is also a favorite topic of many of the examiners. You should remember this one. Okay, a palindrome in DNA, sequence of base pairs that reads the same on both the strands. We know that like DNA is a double stranded, okay, double helix model. We know, we always study this one. Okay, double helix model, one strand and second strand. One strand reads from 5' to 3', and the other strand reads from the 3' to 5' direction. Okay, so when one a sequence of base pairs. Okay, what do you mean by base pairs? This GAATTC, the reading on that DNA strand is called as, one is called as a base pair. So that pair, that reading from 5' to 3', and on the other strand from 3 to 5, that read having a same on both the strand with orientation is kept with 5' to 3'. This is called as a palindrome. In simple words, आगे से भी वही पढ़ा जाएगा, same letters, और पीछे से भी same आए, उसे कहते हैं palindrome. Okay, so biological palindromes, recognition sequences. A palindrome in DNA is a sequence of base pairs the same on both the strands when orientation is kept 5' to 3 prime. So if we are keeping those strands, then these palindromic sequence are same. It shows, a symmetry within the double stranded DNA, and the orientation is critical. And you should always remember that these palindromes are important. Why we are going to study now, but you should remember what is palindrome. Sometime direct basic what is not a palindrome type of sentences could be asked, and those sentences could be a confusing ones. Common palindromic sequences tested are in the equary, it is GAATTC from the Escherichia coli, and from the hind tree, it is AAGCTT, that is from Haemophilus influenzae, and one SAL1 palindrome is GTC and GSC, that is from Streptomyces albus, and BAMHI, that is GGATCCC, Bacillus coefficients, and PVU1, that is CGATCG, and PST1 CTGCAG. This is such a dry topic. If it is going to if in a question this comes as a match the following, then most of the students might be doing wrong. So remember, these are the common palindromic sequences biology students should remember by heart this to its very thorough level, so that any question from these should be answered very easily for my students. Moving on, let's see how the restriction enzymes are cut. Okay, you can see in the diagram also. Diagram is self-explanatory in nature. Okay, the molecular scissors or the restriction enzyme mechanisms. Historical landmark and overview. Concept first is isolating. Okay, concept is isolating the gene sticky ends. Okay, equine cuts between G and A. Okay, restriction enzyme a molecular scissor, it will cut, and antibiotic resistant gene will be produced. Then we are going to have that producing of the sticky ends, we are going to make that E. coli cell in plasmid extraction. Okay, then linking and integration, and then comes the sticky end and the blunt end concept. Okay, the sticky ends means overhanging single stranded stretches that can form hydrogen bonds. Okay, sticky yani that is going to get stuck, जो लग जाएंगे चिपक जाएंगे उसे कह देंगे sticky. So those are gross words. The biological sense is basically they can form the hydrogen bonds with the complementary ends. They are going to form a strong bond, that is called as sticky ends, and the blunt end means obviously the opposite. Okay, so sticky end mechanism and the blunt end. Then we are going to study the cloning and inheritance. So they what does they do actually? They cut away from the center of the palindrome site between the same two bases on the opposite strands. This produces sticky ends. Some enzymes produces blunt ends. Example is ECORV. Recognition sequence is GAT-ATC cuts between T and A. Okay, whereas this ECORI cuts between G and A. G and A, and this one ECORV cuts between T and A, and that's how this ECORV produces blunt end, whereas ECORI produces the sticky end. You should remember this. Direct questions could be asked from this topic. Moving on, examiner's trap. ECORI cuts between G and A in GAATTC, producing sticky ends, and ECORV produces blunt ends. Okay, and it cuts between T and A. You should remember that one. A previously asked question in NEET is which restriction enzyme produces blunt ends? That is ECORV, blunt ends. Okay, and it cuts between T and A. Why don't bacteria cut their own DNA? Okay, such type of reason he is going to ask, like why and how. In assertion reasons and statements also, you should know bacterial DNA is methylated at the recognition sequences by methylase, protecting it from its own restriction enzymes. So the presence of methylase enzyme is helping the bacteria so that its own restriction enzyme is not going to cut at that recognition sites. यानी ये जो methylase enzyme है वो उसको छुपा देगा, ताकि restriction enzymes जो है ना खुद के enzymes फिर खुद के DNA को वो नहीं कट करेंगे. Number of restriction enzymes, fact-based question, over 900 isolated from more than 230 strains of bacteria. Let's integrate this topic with the previous year question. Restriction endonucleases are used in genetic engineering because they cut at a specific base sequence. That is the reason. Which of the following is not true about restriction enzymes? As I have told you, true and false statement type questions are going to come. So they cut only one strand, that is a false, so don't fall that one. They cut both the strands and that to a specific location, which we have seen that locations also. Which palindromic sequence is recognized by Ikori? I told you, you have to by heart this as it is. There is no other option. You have to by heart. Ikori cuts the 5 prime to 3 prime orientation GAATTC. Okay, and the cut point also we have studied, that is GNA. Which of the following represents a palindromic sequence? You should know that palindrome. From both the strands should read same. So when kept in both strand imaginary way, both should read a same sequence of base pair, that is called as palindromic sequence. So these are the some practice questions. Moving on. Separation and isolation of the DNA fragments, gel electrophoresis. Now, for any biotechnology process, obviously we are dealing with genes, we are dealing with DNA. We are not only just cutting it, we are also separating it, and we are also isolating it, and then we are also introducing that into a different medium or host. So we should know specific process that is called as gel electrophoresis. Okay, not only for NEET exam, but in the MBBS also, gel electrophoresis is a very important topic. Always this topic is going to be with you. Okay, so let's study first of all what is gel electrophoresis. Okay, the principle behind the gel electrophoresis is DNA fragments are negatively charged. They migrate towards anode. As simple as that one. Okay. The electrical charge of the DNA fragment is negative, and they are going to go towards anode. We are going to utilize this principle. The principle is not decided by us. Method is decided by us, but the principle is naturally. Okay. So naturally DNA fragments are negatively charged, and due to phosphate backbone, the reason is being given here. Assertion reason question can be expected, and they migrate towards anode, positive electrode under electrical field. So agarose gel acts as a molecular sieve. Sieve is something which you are going to filter, okay, a layer or a structure which is going to filter according to the size of the substances, so that the desirable substances or the smaller size substances are going to be present down, and the undesirable will be present on that one sieve, and we can throw it away. That is called as sieve. Okay, in Hindi and in local language it is called as chhanni basically. Anyways, agarose gel, natural polymer from a seaweed, okay, acts as a molecular sieve. Smaller fragments move farther, they migrate faster through the gel. So this is the principle that we are going to use. Okay, I'm going to have a complete understanding of the process also. So it has three steps. Okay, let's directly have three steps. One is separation and isolation of DNA fragments. Two is gel electrophoresis visualization, and three is elution and purification. Okay, first step is separation and isolation. Okay, then visualization. Okay, then comes the elution and purification. Let's see what are those one by one. Okay, so separation and isolation of DNA fragments. Principle of separation is DNA fragments are negatively charged because of the phosphate backbone. So they migrate towards the anode, okay, positive electrode. Now, after that, when we make that fragments filtered through the agarose gel, because it acts as a sieve, then obviously the smaller fragments move farther, migrate faster, and the larger will be slower. And that's how you are going to separate that fragment. Then you are going to have visualization. Okay, how are we going to visualize? This is very important. We are going to use ethidium bromide. Very important. The name and ETBR, the abbreviation, the process in which it is used, and the step where it is used has been asked multiple times. Okay, so in order to visualize, we are going to use ethidium bromide staining. Okay, so staining and UV exposure. Okay, cannot see DNA in visible light without any staining. So we are going to use this staining. After staining, after exposure to UV radiation, the DNA bands appear bright orange. Okay, and if that bright orange is being seen under that UV radiation, then we will understand that that is a that DNA fragment. So the visualization is helped by ethidium bromide. Then comes the elution and purification. Purified DNA used for the recombinant DNA construction. Band elution and DNA extraction. Okay. Elution is, in simple words, the elution means the separated bands are cut out from the gel. Okay. Gel cut. Simple word. Gel cut. Then comes the purification so that we can use it for recombinant DNA construction. This is called as gel electrophoresis. Okay. Visualization. DNA is stained with ethidium bromide, ETBr. After exposure to UV radiation, DNA bands appear bright orange. Cannot see DNA in visible light without staining. Elution means the separated bands are cut out from the gel and DNA is extracted. This purified DNA is used for constructing the recombinant DNA. So previous year questions, let's see previous year question and let's integrate that with, just now we have read that topic, let's integrate with this topic. DNA fragments are separated by which technique? Gel electrophoresis. Okay. The word itself tells there is a gel, agarose gel, which acts as a sieve. Okay. And electrical charges we are going to phoresis, there is a differentiation between the fragments depending upon their size and weight. Okay. DNA fragments are charged. Yes, they are charged negatively. Ethidium bromide stained DNA on UV exposure shows color. Yes, it does show color, and that color is bright orange. That is how we are going to identify these DNA fragments. Which statement about the gel electrophoresis is incorrect? He has asked this one. Okay, separated DNA fragments can be directly seen under UV radiation is incorrect statement. No, we need UV radiation, but after staining only we need the UV radiation is incorrect because staining with ETBr is required. After ETBr staining, we are going to have UV radiation. Okay, so see that's a subtle trap there. Smaller DNA fragments are observed near anode positive electrode, and agarose is extracted from the seaweeds. So these were the previous year questions, and almost all questions will be asked from this area only. It will either it will take into a statement or match the following or direct fact-based recall question. Third topic is cloning vectors. Okay, you know what is cloning? It is like lab-based reproduction. Simple that way. From one, we are going to make two. From two, we are going to make four in the lab. That is called as cloning. Okay. But we have studied this in the bacteriophages and bacteria. Okay. So a DNA molecule, a plasmid bacteriophage that can carry foreign DNA into a host cell and replicate. It will carry the foreign DNA and it will divide that DNA into multiple times, and that is how we are going to study the cloning vectors. Okay, essential features and PVR322 anatomy is very important. Most of the questions have been already asked. Let's see a complete overview. We can see the diagram there. It is very self-explanatory diagram. Focus on the diagram only. A DNA molecule from plasmid and a bacteriophage that can carry something which is carrying is called as vector. वो चीज जो carry करती है उसे vector कहते हैं, which has a direction also and which has a magnitude also. So that can carry foreign DNA in a host cell and replicate there is called as a vector. Okay. For in order to tell that this is a vector, there are four mandatory essential features. Okay. One is ORI, origin of replication. Second is selectable marker, and third is cloning sites, and four is size and replicability. Let's see what are those four mandatory essential features. Okay, it has to have ORI. Replication starts here and controls the copy number. If there is no ORI, then the copy number will not be controlled. Okay. So we need ORI, origin of replication, in order to start the replication, one, and also in order to control the copy of numbers we are going to copy, and that is divide there. Okay. Selectable marker, we are going to identify and eliminate the non-transformers. Okay, we are going to select, it means we are going to select what we are desiring, and obviously the undesired or the non-transformers are going to be separated. Okay, cloning sites, we are going to have restriction sites. Very few, preferably single site is seen. Multiple sites generate equalized cells do not carry these things. Okay, these cloning sites. Size and replicability, small and able to replicate inside the host. If these four features are available, then we can tell that, that's a cloning vector. Okay, let's see PBR 322. This number, this thing, this concept is highly, highly important. Not only in the MBBS, you are going to study the PBR 322 in MBBS, in B Pharmacy, in biotechnology, all along the science of biology. You are going to see this one. It is very important. It is important to NEET, obviously, but it is important for your life actually. Okay. The most important cloning vector for NEET is PBR 322. Okay. First of all, just if somebody asks what is PBR 322, like one line answer, the PBR 322 is it is a code for replication of proteins. It codes for replication proteins. It codes for replication proteins. Okay, that is important for you. You have to understand that one. There are many parts for that one. Okay, one part is TET-R, another part is AMP-R, and one more part is ROP. Okay, we are going to break down that one. AMP-R means ampicillin resistance gene. Repeat with me three times. AMP-R means ampicillin resistance gene. AMP-R means ampicillin resistance gene. AMP-R means ampicillin resistance gene. Okay, similarly, repeat all the other two parts also three times. Stop the video and you will read on. Okay, this is ampicillin resistant gene and it contains sites like PSTL and PVUA. We are going to study that one in detail. Now just focus on the structure. Okay. The other part is TETR, tetracycline resistance gene. Okay. And it has part like BAMHI, SALA, and we are going to have unique restriction sites also. That is TETR. And we are going to have ROP. Okay, we are going to study it second slide. Don't worry, just focus on the image. Mechanisms, copy number and cloning site requirement. A copy number controls are critical. Cloning site, single unique site, we have to have that one. We are going to study in the second slide. Essential features of a cloning vector. All four are mandatory. Without, if we miss one, obviously that cannot be called or used as a cloning vector. Okay. Origin of replication. It is mandatory for in order to start the replication, and also it is mandatory to control the copy of number. Okay. A question could come like what is the need or what is the requirement, what is the function of ORI? It is not only functioning for the replication starting point, but also a control copy number. High copy number vectors are used if many copies of target DNA are needed. Second criteria, selectable marker, helps identify and eliminate non-transformers. Obviously, we are selecting it with a marker so that what we desire is being taken, which we don't need. We are not focusing on that. Usually, antibiotic resistance genes, ampicillin, tetracycline, kanamycin, and chloramphenicol are used. Normal equalized cells do not carry these resistances. You should remember that one. Cloning sites, also called as restriction sites, should be have very few, preferably single recognition sites for common restriction enzymes. Multiple sites would generate several fragments and complicate the cloning. Okay, in vector PBR322, there are unique restriction sites for HindIII, EcoRe, BAMHI, SAL-I, and PVL1, PSTL, and CLL. We have seen the diagram also. Size and replicability should be small and able to replicate inside the host. PBR322, the most important cloning vector for NEET. ORI stands for origin of replication. ROP, part of that PBR322, is codes for proteins involved in replication of plasmid. AMP-R, ampicillin resistance gene, and TET-R, tetracycline resistance gene. PBR322 is very important. Moving on, memory trick. Okay, this there are many parts now like PST and PVU. Okay, These two parts starts with P and penicillin starts with P. Therefore, it is ampicillin resistant. Ampicillin is actually a type of penicillin. Okay, BAM-HI and SAL1B stand and S for tetracycline, T for tet, but B and S are inside the tet R. Okay, so that a kind of small memory trick. If not, you can directly by heart also. Selectable marker strategies. What are the strategies that you are going to have in order to have the selectable markers? Okay. Antibiotic inactivation method. Okay. Two plasmid method is cumbersome. Insert a foreign DNA at the BAM-HI site inside the TETR. Then TETR becomes inactive. AMP-R remains active. Transformants grown on ampicillin medium. All transformants survive. Then replicated on tetracycline medium. Recombinants with insert in tet are die on tetracycline, whereas the non-recombinants without insert grow on tetracycline. You can see the diagram there. Okay. Antibiotic inactivation method. It means obviously we have seen that in PBR322 there are two sites. One is TETR site and another is AMPR site. So we are going to inactivate one site so that we can have only one site active and we can see that type only to grow. Okay. So TETR and there is AMPR. Okay. After that one insert foreign DNA at BAM-HI site. TET-R is inactive and AMP-R is active. All transformants will survive. Okay. Then we are going to have the replica plating. Then the recombinants. Okay. In the TET-R, they die, whereas in the other one, okay, they grow. You have to understand this one. Okay. Remember the diagram also. Important points. Process will be getting as questions, or at least true or false will be also a part of questions. Selectable marker strategy, insertional inactivation using beta galactosidase, blue-white screening simpler. Okay, so another method of this strategy is insertion. Insert, it means imbibing it, inserting it, attaching it. Inactivation using the beta galactosidase. What is happening here? Vector contains the LAGZ gene. LAGZ gene codes for beta galactosidase. You should remember sometime direct fact-based questions are asked. LAGZ gene codes for beta galactosidase. Insert the foreign DNA inside the LAGZ, then it converts into insertional inactivation of beta galactosidase on medium containing X-gel chromogenic substrate, non-recombinant colonies, active B-gal blue and recombinant colonies, inactive B-gal white. This is the most commonly used method in the modern cloning. Okay, so in the modern cloning, the method we are going to have for the selectable marker is insertional inactivation using beta galactosidase, most common. PYQ integration, previous year question. Let's see what type of questions are being asked of him. The two antibiotic resistance genes on PBR322 are ampicillin tetracycline, AMP-R or TET-R. Okay. Ligation of foreign DNA at which site will result in loss of tetracycline resistance, BAM-HI. Insertion at the PSTL site results in loss of ampicillin resistance. Blue-white selection is seen in blue colonies are non-recombinant active VGAL, that is beta-galactosidase. White colonies are recombinant, that is insertional inactivation. What is not a cloning vector? Probe is not a cloning vector. Probe is a labeled nucleic acid for detection, not a vector. Probe is used for detection. It is not a vector. Vectors for plants and animals. Okay, most commonly used. Okay, most commonly used vectors for plant is from agrobacterium tumefaciens. Okay, Ti plasmid, that is tumor inducing plasmid. Ti plasmid, Ti stands for tumor inducing. Direct question could be asked for dicot plants. The tDNA region is transferred to plant genome. Disarmed Ti plasmids are now used as cloning vectors. One has to remember that commonly in plants we used the Ti plasmid, the tDNA region, as from the agrobacterium tumefaciens in plants. Okay, and in animals, obviously retrovirus is used to deliver genes into animal cells. Disarmed retroviruses are vectors for gene therapy. You can see the diagram also. This diagram is self-explanatory in nature. But if you see the diagram and if you imagine that properly, it will be easier for you in the exam. PYQ integration. If you have any doubt, you can comment in the comment section. I'm going to reply you there definitely. Commonly used vector for gene transfer in plants is from Agrobacterium tumefaciens. Ti plasmid is found in Agrobacterium. Okay, Ti means Ti stands for tumor inducing. That could be also a question. Retrovirus is used as a vector for introducing DNA into human lymphocytes. Obviously, in animals, obviously, human category comes under animals. Gene therapy for ADA deficiency is also an example we are going to study also. Competent host. How are you going to make cells ready to take up DNA? If you are doing a gene therapy or a recombinant DNA technology, if you are doing any process or diagnosis, okay, how are you going to make that cell ready to take a DNA? How are you going to? That is called as four competent host mechanism. Okay. Why is DNA not taken up easily? DNA is hydrophilic and cannot cross the cell membrane. That is the reason that DNA is not taken up easily. Assertion reason is question can be asked from this line. Okay. Methods to make the host cell competent. How are we going to make the host cell competent in order to take the DNA? Okay. There are four methods. One is calcium chloride treatment plus heat shock. Second is microinjection. Third is bioballistic gene gun. And four is disarmed pathogen vectors. Remember these four methods? He directly four methods could be four options, and any one options he can confuse you, and that could be a question. Okay. Calcium chloride treatment and heat shock. Treat the bacterial cells with specific concentration of calcium. Incubate with the recombinant DNA on ice, brief heat shock it at 42 degrees centigrade, then back on ice, creates pores in cell wall membrane for DNA entry. By doing this process, the cell wall membrane will have small pores and DNA can enter. This is called as calcium chloride treatment with heat shock. Second step is microinjection. Directly inject DNA into nucleus of the animal cell. Okay, directly you have to do that one. Biolistics gene gun, high velocity microparticles of gold or tungsten coated with DNA are bombarded into the plant cells, also called as particle bombardment. Okay, biolistics means particle bombardment. Gene gun device, DNA coated with gold or tungsten is used. Fourth method is disarmed pathogen vectors. Use modified agrobacterium or retroviruses to deliver the DNA. That is disarmed pathogen vectors. We have already studied about vectors. Microinjection, biolistics. I have explained this one in the image. You can stop and read it. Disarmed pathogen vectors. Previous year question integration. In gene gun method, microparticles of which metal are used? Tungsten or gold? Obviously both. Biolistics is suitable for transformation of plant cells. Calcium chloride treatment is used for making bacterial cells competent. Let's see the process of DNA technology. Okay, recombinant DNA technology, rDNA technology. Okay, obviously we are going to study step by step. First step is isolation of genetic material, that is DNA. How are we going to isolate? Cell wall is broken using specific enzymes. Then we are going to use lysozyme from bacteria, cellulase plant cells, and chitinase for fungi. And that's how we are going to break the cell wall. For different organisms, we are going to use different. For bacteria, lysozyme. For plant cell, cellulase. And for fungi, it is chitinase. Okay. Then we are going to add the RNAs to remove the RNA. Then we are going to add protease to remove protein, and then other macromolecules removed by appropriate treatments like polysaccharides and lipids and other impurities. Chilled ethanol is added to precipitate out purified DNA. So we are breaking down the bacteria, we are differentiating the RNAs, we are differentiating the proteins in these steps. Finally, we are going to add the chilled ethanol, and this chilled ethanol is going to precipitate. Okay, it is going to make that settle down, and that's how you can find that those fine threads which are settled after the treatment with the chilled ethanol are nothing but that DNA precipitates. Okay, so this is the first step, isolation of genetic material. Previous year question, okay, which enzyme is used to isolate DNA from fungal cells? Chitinase. Isolation of DNA from plant cell requires cellulase. Addition of chilled ethanol precipitates DNA, and chilled ethanol is used as added to precipitate DNA. Cutting of DNA at specific location. Now we have separated DNA, we have got DNA. Now we have to cut at the specific location. Okay, purified DNA is incubated with restriction enzyme under optimal conditions. Vector DNA is also cut with the same restriction enzyme. The cut ends are sticky or complementary, essential for ligation. Sticky ends are going to get bonded. That is called as ligation. So purified DNA, then comes restriction enzymes, then comes the vector DNA. Okay, this is the cutting of DNA at specific location. Donor DNA, we are going to add restriction enzyme, then there it is going to cut and sticky ends are produced. Then that cutting mechanism, after with the sticky ends, we are going to add with this vector DNA. So remember this. You can see the diagram also. Diagram are very clear. If you see the diagram only, you can imagine, and that imagination is going to help you by heart also easily, and you will not forget. PYQ integration, previous year question. Recombinant DNA molecule can be created normally by cutting vector and source DNA with same restriction enzyme, HIND2 and HIND2. Unless we cut vector and source DNA with the same restriction enzyme, the recombinant vector cannot be created. Direct statement from NCRT, often asked as a statement based question. Okay, direct statement from NCRT. Read these two statements, stop the video here, read these two statements 5 times, maybe write it down somewhere. Step 3 of this RDNA technology is amplification of the gene of interest. So after cutting and after identifying, then we are going to amplify. We are going to produce it for multiple times. Okay. How are we going to produce? Through obviously polymerase chain reaction. Okay. In vitro DNA replication that produces billions of copies of the target DNA sequence is called as polymerase chain reaction. Okay. Three steps in each PCR cycle. One, denaturation, annealing, and extension. Okay, what is denaturation? At 94 degrees centigrade, double stranded DNA separates into a single strand. Denaturation. Annealing at 50 to 60 degree temperature, the primer, okay, bind to complementary sequences on the template of the strand. Okay, then comes the extension at 72 degrees centigrade. Thermostable DNA polymerase, that is Taq polymerase, adds nucleotides to the 3' end of the primers. So repeat cycle 20 to 35 times, and that's how PCR is going to produce millions of copies. Let's see how many copies. Like number of DNA copies after n cycle is 2 to the power of n, starting from one double stranded molecule. Okay, exponential amplification. Start one DNA molecule, one cycle 2 copies, two cycle 4 copies, three cycle 8 copies, and n cycles 2 to the power of n copies will be produced. Okay, number of cycle and number of copies, that could be a direct question, numerical question. Key enzyme in this PCR amplification is Taq polymerase. Okay, and isolated from the thermophilic bacteria Thermus aquaticus. Okay, and heat stable enzyme Taq polymerase. Direct question could be asked, like which of the following statement is correct, or which of the following statements is incorrect about Taq polymerase. Okay, Taq polymerase enzyme that makes PCR possible. Okay, it is isolated from the thermophilic bacterium Thermus aquaticus. It is a heat stable enzyme. It remains active at 72 degrees centigrade. Okay, and the bacteria is Thermus aquaticus. Okay, role in the PCR, obviously it is going to help with the primer and the Taq polymerase at the 70 degrees centigrades. It is going to help with the replication at the further step. Very easy slide here. PCR application, polymerase chain reaction. It is helpful in DNA amplification, cloning, and for the molecular diagnosis. For example, you have to diagnose a disease in a patient. We are going to like HIV identification, genetic disorders, cancer mutation. These all diagnosis. Diagnosis means identifying the disease by a lab process. Okay, and it is not used for protein purification. Remember that when it is not used for protein purification. Okay, these are the applications of PCR. Correct the sequence of steps in PCR: denaturation, annealing, extension, and the sequence of the temperature also. You should remember that one. Okay. PCR amplifies DNA following the equation 2 to the power of n. Thermostable DNA polymerase used in PCR is isolated from Thermus aquaticus, which is not an application for PCR, purification of isolated protein. We don't do that one. You should remember. If high temperature is not maintained in the beginning of PCR, which step will be affected? Obviously, the first step is affected because we need 94 degrees centigrade at the first step, denaturation. Insertion of the recombinant DNA into the host cell. Now we have got that DNA, we are going to insert in the host cell. Okay, component cells are mixed with the recombinant DNA. Transformants are selected using selectable markers, antibiotic resistance. Only transformed cells grow on antibiotic containing medium. What are we going to do in step 4 and step 5? Let's see. In step 4, insertion of the recombinant DNA into the host cell happens. Recombinant DNA plus the host cell transformation, selection on the antibiotic medium. After antibiotic medium, transformed cells grow and non-transformed cells do not grow. And only transformed cells grow, then we are going to have that competent cell, recombinant DNA entry, transformants, selection, then survival of the transformant. This is the sequence. Step 5, obtaining the foreign gene product. Recombinant DNA, we are going to help with cloning in the host cell. Then that expression of the foreign gene happens. Then the recombinant protein is produced. That protein produced by a heterologous host is called as a recombinant protein. Then small scale cultures are converted into a large scale cultures in the bioreactors. Then the recombinant protein at a large scale can be produced. Okay. Step 5, obtaining the foreign gene product. As we have seen in the diagram, this video, you can stop the video and study, read it. Bioreactors, most commonly used are stirred tank bioreactors. Vessels of 100 to 1000 liters capacity provide optimal growth conditions, like they are going to provide the conditions of temperature, pH values, oxygen, substrate, salts, vitamins, etc. What are the features of the bioreactors? Agitator system. It means for even mixing and oxygen availability, oxygen delivery system, and foam control system. Okay, the sparger for air bubbles and foam breaker, temperature and pH control system, and sampling ports. All these are the parts of bioreactors. Parts of the bioreactors 1, 2, 3, 4, 5, that are direct questions. Optimal growth conditions are temperature, pH, oxygen, substrate, and salts and vitamins. That is also direct fact-based question. What is the capacity of this bioreactor? 100 to 1000. What are the features? Is agitator system, oxygen delivery system, foam control system, and temperature and pH control system. So these all are important when it comes to bioreactors. Downstream processing is the last step when it comes to this technology. Okay, after biosynthetic stage, the product is subjected to a series of processes. One is separation, okay, removal of cells and impurities. Then comes purification, isolation and purification of the desired product. Then comes preservation, stability and shelf life maintenance of that product. We are going to preserve it. Then formulation for marketing, preparation of final product for marketing. That is upstream process. Expression of recombinant DNA is not a part of downstream processing. Okay, expression is not a process. Okay, downstream process is all about how we are going to make that product ready for the market. That is downstream. Okay, the process of separation and purification of expressed protein before marketing is called as downstream processing, which is not a component of downstream processing. Expression is not. Frequently confused terms, let's see. Exonucleases removes nucleotides from ends of the DNA. Remember, this is all a revision video. Endonuclease cuts DNA at specific internal positions. Restriction enzyme, a type of endonuclease class 2 that recognizes specific sequences. You have to read this multiple times. Stop the video here. Read this 3 to 4 times. Direct match the following questions could be expected. Okay, a restriction enzyme, a type of endonuclease class 2 that recognizes specific sequences. DNA ligase joins two DNA fragments, seal needs used after restriction cutting Tag polymerase, thermostable DNA polymerase for PCR, probe single stranded DNA or RNA tagged with a radioactive molecule for hybridization. So probe single-stranded DNA or RNA tagged with radioactive molecule for hybridization. Selectable marker gene that helps identify transformants. See, after studying all these processes, few of the things are used in one process, and the other things are used in other process. So during the exam, there's a chance that you get confused. So these are confused terms. These are NEET favorite. Okay, and reporter gene, example like gene whose product is easily assayed for monitoring the transformation. Repeated questions are being asked from this term. Stop the video here, write it down, or listen to this video like multiple times. Previous year question: enzyme catalyzing removal of nucleotides from ends of DNA is ends of DNA exonuclease. Joins two DNA molecules ligase. Single stranded nucleic acid tagged with radioactive molecule probe. Gene whose expression helps identify transformed cell selectable marker. Which of the following is not essential for gene cloning? DNA mutase and recombinase is not needed for cloning. Okay, essential are restriction enzymes, DNA ligase, and DNA polymerase. Miscellaneous important points, common concepts. Like there are some concepts in the NCRT which are not like bold, or which are not italics, which are not boxed, which don't have diagrams, etc. They are the corner words or statements in the paragraphs, ending chapters. Going to give you that also, which are sometimes important. Okay, what is a palindromic sequence? Direct definition: a sequence of base pairs that reads the same on both the strands when orientation is kept from 5 prime to 3 prime. Example, this. Why do restriction enzymes not cut bacterial own DNA? Because they are methylated. Okay. What is the role of the ROP gene in PBRMG22? It codes for proteins involved in replication of the plasmid. What is the function of ORI? Controls copy number and initiates, starts the replication. Can a restriction enzyme cut a DNA molecule that is not methylated? Yes. If not methylated, obviously it can cut. What is the difference between a cosmid and a plasmid? Cosmid is a hybrid vector, plasmid plus phage cos sites that can carry large inserts, 45 kilobytes of the length of the base pairs than the plasmids. For human genome sequencing, BAC, that is bacterial artificial chromosome, and YAC, that is yeast artificial chromosome, are used when it comes to cosmid and plasmid in humans. Commonly used vectors for human genome sequencing are BAC and YAC. Which vector can clone only a small fragment of DNA? Plasmid. Predicted. Expected questions are assertion reasons. Okay. Restriction enzymes produce sticky ends. Reason: they cut the DNA strand a little away from the center of the palindromic site. It is true. Both the statements are true, and that is a correct explanation. That is called as sticky. Statement based: which of the following is or are correct? Required for PCR: DNA template, primers, stack polymerase, deoxynucleotides, and buffer. Not required are in the PCR is ligase and restriction enzyme. Sequence based: arrange the steps of rDNA technology: isolation of DNA, cutting with restriction enzyme, ligation into the vector, transformation, and selection. The correct sequence is presented here. The question could be interchanged. Match the following. Okay, column one, the questions could come into columns. Okay, just stop the video, write it down, and by heart. These are the expected questions in the NEET. Diagram based questions: a figure of PBR322 he will give, and he is going to ask you about ORI, origin of replication. He's going to ask you about rope. He's going to ask you about AMP-R and TET-R and restriction sites, and which site is for PSTL and which for BAM-HI. He is going to ask. The diagram which we have shown in the previous slide is very important, and direct diagram based questions are expected in the future NEET. Biotechnology and applications, the 10th chapter. Okay, the examiner's blueprint, as in the 9th chapter also we have seen which topics are very important: bt toxin and bt cotton, pest resistant, and genetically engineered insulin. These are highly important topics. We are going to study these topics in our classes. Okay, now in the further slides, gene therapy, ADA deficiency, molecular diagnosis, these are very important. Sometimes low topics, like less important topics, are transgenic animals and ethical issues. But they might come, but highly important are the above said. Let's see biotechnological applications in agriculture. Okay, these are three options. Many other options could also he will add. You have to understand that logically. Sometimes questions directly from textbooks, but application is not textbook. Application is your brain, how far it can understand. Okay, three options: agrochemical based agriculture, organic agriculture, genetically engineered crop based agriculture. Okay, these three. Let's see what is tissue culture. Okay, the capacity to generate a whole plant from one cell is called as totipotency. The capacity to generate a whole plant from any cell or explant is called as totipotency. Single cell explant is used in culture, then that culture produces a plantlet, then that plantlet is taken out and put in the soil natural environment, that is totipotency. What is explant? Any part of a plant taken out and grown in test tube under the sterile conditions is called as explant. Okay, from leaf or a stem or a root or shoot tip or flower. That is how this explant is produced. Okay, nutrient medium must provide. What should we provide in the nutrient medium in order to produce this tissue culture? Carbon source sucrose we will provide, inorganic salts of sodium, potassium, and nitrogen, calcium, magnesium, iron we will provide. Vitamins are provided like vitamins B6, B2, B12, and vitamin K, B3, A, D, E, and C. Amino acids will provide. Growth regulators like auxins and cytokinins also be provided. Explant, sterile culture, multiplication, rooting, then direct the plant is produced. This is very important. Stop the video here, read it 3 to 4 times. You will be perfect, and any question from this topic will be easier for you to answer. Tissue culture and micropropagation. Micropropagation means production of thousands of plants. Okay, propagation many. Okay, through tissue, because tissue culture is done at a micro level in a test tube, so this is called as micropropagation. Okay, production of thousands of plants through tissue culture is called as micropropagation. Okay, we are going to take mother plant, we are going to produce an explant of that mother plant through tissue culture. We are going to multiply it through mass propagation, then thousands of plants are produced. All plants are genetically identical to the original plant called soma clones. Okay, so in the micropropagation, all plants are similar to the mother plant. Okay, virus-free plants can be obtained by culturing merisstems, apical and auxiliary, because merisstems are free of viruses. Apical merisstems. The tip part is called as apical meristem and auxiliary meristem. These two, apical meristem and auxiliary meristems, don't have viruses. So virus-free plants can be produced if we are going to prepare the explant through tissue culture from the apical meristem or the auxiliary meristem. Very important question for NEET exam. Okay, examples are banana, sugarcane, potato. Micropropagation ensures rapid multiplication, uniformity, and healthy products. Okay, somatic hybridization. Somatic, soma means body. Protoplast cells with cell wall are removed from two different plant varieties, are fused to form hybrid protoplast, which are grown to form a new plant. Okay, that is called as somatic. Protoplast from two different plant varieties, okay, are used to form a hybrid protoplast, which are grown to form a new plant. Okay, parent plants. First step is having a parent plant. Second step is isolation of the protoplast of each plant. Third step is fusion of the protoplast to produce a hybrid protoplast. Fourth step is culture of the hybrid protoplast. Fifth step is development of a new plant. This is called as somatic hybrid. Okay. Genetically modified organisms we have studied now our previous slides also. Tolerance to abiotic stress. Okay, we are producing these modifications because in order to survive, in order to live, in order to have uh, protection from the stress from the environment. Okay, the main benefits of the GM plants are: one, tolerance to abiotic stresses, cold, extreme cold, drought conditions, salt conditions, and heat conditions. From all these stress, these plants will tolerate. That is genetically modified organisms. Okay, reduced reliance on chemical pesticides or pesticides. Okay, pest resistant crops are produced. Healthier crops, less pesticides, reduced post harvest losses. Okay, and less wastage, more savings, increased efficiency of mineral usage, better nutrient uptake, higher use efficiency, enhanced nutritional value. Example, golden rice, vitamin A enriched is golden rice, helps prevent vitamin A deficiency. Golden rice is a genetically modified plant for better health. Okay, it is a gene from maize PSY, gene from bacteria CRTI, and inserted into a rice genome. Then golden rice plant is produced. Golden rice grains rich in vitamin A, beta carotene, helps prevent vitamin A deficiency and improves the health and immunity. This is about genetically modified organism. Stop the video here. Memorize the image. BT toxin and BT cotton. Most important topic. Okay. Bacillus thuringiensis or giensis. Both the pronunciations are seen in the world. Okay. Source is Bacillus thuringiensis, a soil bacterium. Most important is going to produce the BT cotton. Let's see what is this one. Okay. BT stands for the bacteria name. Exist as inactive protoxin inside the bacterium as crystal, protein crystal, protein crystal, protoxin present in the bacterium. Okay, when insect ingest it, the alkaline pH of the insect gut solubilizes the crystals and convert the prototoxin into active toxin. Activated toxin binds to the midgut epithelial cells and create pores and cell swelling and lysis. Then that insect will die. Then from the Bacillus thuringiensis, we are going to take out the prototoxin crystals, then ingestion by insects, then solubilization, then binding to midgut cells and pore formation. These all are going to happen, and ultimately, that is, the insect is going to die. Okay, BT toxin and BT cotton. Source obviously we have studied. Okay, this all process happens. But what are cry-genes? Very important. Cry-genes are gene coding BT toxin is called as cry-genes. Different cry proteins target different insect orders. Okay, like cry1AC and cry2AB control the cotton bollworms. So these BT means they are resistant actually. Okay, and cry1AB controls the corn borer. BT cotton is a transgenic cotton expressing cry1AC and cry2AB genes so that it will protect us from these insect causing cotton bollworms and the corn borers. Okay, stop the video here. Study the image thoroughly and by heart. Previous year questions, very high frequency. BT cotton variety is resistant to insect pests. Okay, no insect can now bite this cotton once it is produced from the BT. What triggers activation of prototoxin to active BT toxin in bollworm? Alkaline pH of the gut of insects. Sequence of action of the BT toxin in BT cotton. So this sequence is already asked previously and he's going to ask again also. Cry1AC gene controls cotton bollworm and Cry1AB gene controls the corn borer. BT toxin does not kill the bacteria because exists as inactive prototoxin. The activated BT toxin binds to midgut epithelial cells and creates the active form of that one and kills the insects. Pest-resistant plants, RNA interference, RNAi, nematode resistance. Any words are synonymous. Any question come with any of the words will be the same question. Okay. The pathogen here we are talking is Melodogyne incognita. Remember the pathogen name. A nematode that infects roots of the tobacco plants. Very important topic. Any topic if the question comes from it starts from pest resistant plant, or if the question starts from RNA interference, or it starts from nematode resistance, all these three topics will be the same. Okay, the pathogen is Melodogyne dogain incognita. What happens here is RNAi mechanism. Let's see what happens is, okay. Double stranded RNA, that is dsRNA, complementary to specific mRNA, is present in the cell. Okay. Dicer enzyme recognizes the dsRNA and cleaves it into small interfering RNAs, small RNAs, small interfering siRNA by dicer enzyme. One strand of siRNA, guide strand, is incorporated into RISC complex. The guide strand in RISC binds to complementary target mRNA. Then argonate protein in RISC cleaves the target mRNA at the binding site. The cleaved mRNA is degraded by cellular nucleases. Read this sequence like 10 times. Sequence-based questions are coming here. Okay. Source of the complementary RNA, infection by RNA viruses, viral RNA or the double-stranded RNA formed during viral replication can trigger the RNAi. Then complementary RNAs may originate from second is transposon, that is mobile genetic elements which are moving. Okay, double-stranded RNA from the transposon transcripts can form double-stranded RNA and trigger the RNAi. Okay, what happens here is it silences the essential genes of that pathogen. So these things are going to silence the essential genes of that pathogen, that is that nematode, and that's how it will die. How transgenic plants are made resistant. Okay, how they are made resistant? Using agrobacterium vector, nematode specific genes are introduced into the host plant. Okay, we are going to use the vector. We have studied in the vector chapter in the slide that we are for plants the common vector is agrobacterium. We are going to introduce the genes in the host plant of the nematode. The transgene produces both sense and antisense. They form double-stranded DNA. The double-stranded DNA initiates the RNAi interference and silences the specific mRNA of the nematode. If that nematode is going inside the plant, nematode cannot survive in the transgenic host. इस तरह से that nematode is going to die in the plant. Previous questions. RNA interference involves silencing of specific mRNA due to the complementary double-stranded RNA. RNAi is used in developing plants resistant to nematodes. Transposons can be used in gene silencing RNAi. Which part of tobacco is infected by Meloidogain incognita root? Biotechnological applications in medicine. This is very easy topic actually. It means in the medical line, in hospitals and labs and other places, what is the use of these biotechnological applications? Okay, recombinant therapeutics advantages, mass production of safe and effective drugs, no unwanted immunological responses, and more than 30 recombinant therapeutics approved worldwide, 12 in India. Genetically engineered insulin, 100% expected question. The word itself is also explaining. We are producing the insulin. Genetically, we are producing the insulin. Naturally, in our human body, the insulin is produced. But in diabetes, especially type 1 diabetes, the insulin insensitivity occurs. Okay, or in pancreatic cancers, insulin production is obstructed. So, on that type of diabetic patients, we are going to have this insulin used, and this is genetically engineered insulin called as humulin. Okay, problem with animal insulin from cattle or pig pancreases can cause allergy, so we are going to produce from human itself. Okay, human insulin contains two polypeptide chains. You can see the diagram also. Okay, A chain contains 21 amino acids and the B chain contains 30 amino acids. Okay, it has double sulfide bridge. N stands for amino acids and C stands for carbonyl carboxyl terminal. Okay, why are we not using animal insulins? Because more allergy is being seen. Then chains are linked with disulfide bridges. That is a fact-based question that is expected. Insulin is synthesized as prohormone, that is proinsulin. Proinsulin contains an extra stretch called C-peptide. Previously, in the previous year, what is C-peptide? Where do you see that type of questions have already been asked? Okay, beta cells of the pancreas Pre-proinsulin contains signal peptide. Signal peptide removed, then it becomes proinsulin with which has C peptide. Then C peptide is removed, then mature insulin is produced. Okay, it is identical to human insulin. No allergic reaction is seen, and it is safer and more effective. Recombinant production of insulin, humulin. Okay, two DNA sequences corresponding to A chain and B chain were prepared separately, introduced into plasmids of E. coli. Chains A and B were produced separately, extracted, and combined by creating disulfide bridges. This mature insulin without C peptide is called as humulin. Stop the video here. Study this image like 10 times, the sequence, the diagram, and try to memorize the image, and every question can be answered easily. First, human hormone produced by rDNA technology, insulin. The two polypeptides of human insulin are linked by disulfide bridges. Which of the following is true for genetically engineered insulin? Correct statements: A peptide and B peptide chains produced separately in E. coli. True. Extracted and combined by disulfide bonds. True. Incorrect are: it does not contain C peptide. Animal insulin may cause allergy. Okay. Proinsulin has extra C peptide. Mature insulin lacks C peptide. Gene therapy, the word itself tells you that therapy means treatment. Okay, by modification of a gene or a correction of a gene defect, by delivery of a normal gene into the individual cells or tissues to compensate for the non-functional gene is called as gene therapy. Okay, first clinical gene therapy happened in 1990 for adenosine deaminase, that is ADA deficiency. Okay, adenosine deaminase. Without these adenosine deaminase, severe combined immunodeficiency. It means there is immune power deficiency, so that that child will have like multiple infections, continuous back-to-back infections. ADA deficiency causes defective ADA enzyme, accumulation of toxic metabolites, failure of the immune system, and recurrent continuous back-to-back infections have are seen in that type of patients. Okay, conventional treatment, what we do is a healthy donor bone marrow cells transplant into the patient, reconstitution of immune system. But now we are replacing in with the gene therapy. Okay, gene therapy for ADA deficiency, child, weak immune system, recurrent infections, lymphocytes isolated from the blood, cell culture has been done, then retroviral vector, as we have studied. For plants we use that bacterium, agrobacterium vector, and in animals, in humans we use retrovirus. Gene insertion happens, then genetically engineered lymphocytes are immune function restored, then that is infused to the plant patient, and the immunity is restored. Stop the video here, read the whole picture step by step. First clinical gene therapy was given for ADA deficiency. Which vector is used to introduce functional ADA gene into lymphocyte retrovirus? Why does the patient require periodic infusion of genetically engineered lymphocytes? Because they are not immortal cells. Gene therapy allows correction of a gene defect. Stop the video, study the previous year questions, then go back to previous video, and you on your own form some questions. Molecular diagnosis. Diagnosis means identification of diseases. And in order to identify, we are going at the molecular level. Okay. Limitation of the conventional methods like normal blood sample or normal urine sample analysis is having limits to identify. We need to go deep. One is PCR, two is ELISA. What is PCR? We have already studied this one, very easy. Polymerase chain reaction. ELISA, enzyme linked immunosorbent assay. Okay. Stop the video here, read the statement. Direct statement based questions are coming from this topic. DNA RNA probe, single stranded DNA or RNA tagged with a radioactive molecule is acting as a probe. Probe only helps for identification. Okay. Detect by autoradiography. Mutated gene with non-complementary sequence will not hybridize, so no band on the film, and that's how you identify. Molecular diagnosis, conventional methods are not working, so we use PCR, ELISA, DNA, RNA probes. Previous year questions. ELISA is based on antigen-antibody interaction. Which technique serves the purpose of early diagnosis? PCR, ELISA, and RDN technology. Key reagent in ELISA is alkaline phosphatase, and PCR is used to detect HIV and genetic mutations. Transgenic animals. This is very easy topic. I'm just going to read this topic. You yourself stop the video and read 3 times. I'm just going to read it. It is very easy. Transgenic animals. Definition: animals that have had their DNA manipulated to possess and express an extra foreign gene. Over 95% of all the existing transgenic animals are mice. Purposes of producing transgenic animals: study normal physiology and development. Example: study of insulin like growth factor. Study disease models of for cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Biological products, medicines, example, alpha-1 antitrypsin for emphysema can be produced. Transgenic cow Rosie, produced in 1997, produced human lactalbumin in milk, 2.4 gram per liter. Her milk was nutritionally more balanced for human babies than natural cow milk. Vaccine safety testing: transgenic mice used to test safety of polio vaccine can replace monkeys. Chemical safety testing, toxicity testing: transgenic animals with increased sensitivity to toxins give faster results. I'm telling you, this topic is very easy. Just stop the video, read this one. Apart from this one, you will not need anything for your NEET exam. PYQ integration: maximum number of existing transgenic animals are mice. Milk of the transgenic cow Rosie contained human alpha-lactalbumin. Transgenic animals are used to test the safety of which vaccine? Polio vaccine. Transgenic cow Rosie was produced in which year? 1997. Genetic engineering approval committee: Indian government body that makes decisions regarding validity of the genetically modified organisms and research and safety of introducing the GM organisms for public services. The function of that committee: biopiracy, use of bioresources by multinational companies without proper authorization or compensatory payment from the countries or the people concerned, is called as biopiracy. Stop the video and by heart the definitions. Ethical issues, examples of biopiracy: Basmati rice patent of 1997. US company patented new variety derived from Indian farmers' varieties. India has nearly 2 lakh rice varieties, 27 documented Basmati varieties. Turmeric and neem attempts to patent their medicinal uses. Indian Patents Bill Second Amendment addresses issues of patent terms, emergency provisions, research and development. Patent means, for example, I have discovered one variety, then I'll name that, I'll register with my name, and that type of variety will be registered with my name only, and I will have almost all the rights or minimum rights to use that one, how to use that one, so that other people cannot use, and that's how you earn money also. Which body of the Indian government regulates GM research safety? GEAC, Genetic Engineering Approval Committee. Biopiracy refers to unauthorized use of bioresources without compensatory payment. What is golden rice? A transgenic rice variety engineered to produce beta carotene, provitamin A in the grains, giving them a golden yellow color. Genes introduced from daffodil or maize and a bacterium. Purpose is to combat the vitamin A deficiency causes night blindness and other issues in developing countries. What is the difference between sense RNA and antisense RNA? Sense RNA is the mRNA that carries codons for translation. Antisense RNA is complementary to mRNA, binds to it and prevents translation. So anti it prevents, and sense RNA it has codons for translation. What is C-peptide? Extra stretch present in the proinsulin is C-peptide, removed during maturation, not present in the mature insulin. Its absence is used to identify the recombinant insulin. Why is Agrobacterium tumefaciens called nature's genetic engineer? Because it naturally transfers the tDNA from its plasmid into plant cells, causing crown gall tumors. This property is exploited to deliver the foreign genes. What is the difference between ex situ and in situ conservation? XC2 means conservation outside a natural habitat. Example, botanical gardens, gene banks, cryopreservation. Cryopreservation means preservation of gametes in viable and fertile condition for longer period at lower temperatures. What is protoplast? A plant cell with its cell wall removed, surrounded by only plasma membrane, used in somatic hybridization. We have studied in the previous slides. What is micropropagation? We have already studied. What is elution? We have studied. Just stop the video and read it again and again. What is function of the ethidium bromide? We know that it intercalates into DNA and fluoresces. It means shines under the UV light, allowing the visualization of the DNA band during the DNA segment separation. Why is stacked polymerase used in PSR? Because it is thermostable, remains active at 72 degrees centigrade, and does not denature during the denaturation step at the 94 degrees centigrade at the first step. What is selectable marker in PBR322? AMP-R, TET-R, insertion in TET-R, that is BMHI, and SAL1 gives rise to TET-R lost, and insertion in the AMP-R makes the AMP-R lost. We have already studied in our previous slides which restriction site is present in TET-R of PBR322, BAM, H1, and SAL1. Which restriction site is present in AMP-R of the PBR322, PSTL, and PVUL. These are the expected questions. Stop the video and study multiple times these expected questions in the future NEET. This is very important. Stop here. Again, stop the video here and study these questions. These are the expected questions for future NEET. Pro tip, from which chapter, what are we going to expect? Okay, stop the video. We have studied all these topics in our previous slide. I'm just doing a mop up here. Stop the video and study on your own. You are now ready to solve any NEET question from biotechnology. I'm 100% sure that if like 6 to 7 or 8 questions are coming from biotechnology, 32 marks are yours. 100% you are going to solve all the biology questions pertaining to biotechnology if you watch this video multiple times. All the very best. Let's meet in our next chapter of evolution and ecology. Thank you.